Welcome to MOOC Interactomics course. Kinetic analysis of antibodies and other proteins is critical to characterization of molecules. In today's lecture, we will use biolayer interferometry BLI technique to perform a kinetics interaction study between mouse monoclonal antibody and protein A. Here protein A will be immobilized on a matrix at the tip of a fiber optic sensor. The binding between the immobilized ligand and the analyte. For example, the mouse monoclonal antibody will produce a change in optical thickness at the tip and resulting in a wavelength shift proportional to binding. The data acquisition and data analysis will be demonstrated, followed by determination of interaction kinetics between the analyte and the ligand. So, let us start the lab experimental session now.
Myself Sushilendra Vaidya, application specialist for the FortiBio Pol Corporation. Just now in the last talk, Dr. Shenglan Kao spoke about the biolayer interferometry and the applications. Now I am going to demonstrate how this biolayer interferometry works. Now I am going to demonstrate the kinetic studies of the monoclonal antibodies, that is the mouse monoclonal antibody to the protein A ligand. This is the Pol FortiBio octet red 96 instruments. If you look at inside, this is, we call it as the optical head box. The optical head box consists of a spectrophotometer as well as the channels, or we call it as a manifold. This is 8 channel manifold, so it can take up the 8 sensors. So here if you look at, this is the sensor compartment, this is, we call it as a sample compartment. Now you can see this is, we call it as a sensor holder or sensor manifold. So here we can put it this sensor in this fashion, and then we can put it this as a sample. So this is the 96 well plate format. We can analyze 96 sample in one go. If you look at this is works on the principle of the dip and read as well as the biolayer interferometry. You know about the principle behind the biolayer interferometry in the last talk. Here it consists of a white light, as if you look at here, this is called as a white light. You can able to see here, this is the light emitting diode. We are passing the light emitting diode, the light passed through the sensor. If you look at this is the sensor. It is actually made up of the glass. It consists of a fiber optic inside, and the end at the holder part, this is made up of the plastic. So inside there is a fiber optic. At the tip of the sensor we are putting ligand of our interest. So we are passing a white light get reflected back. It is a reflection based phenomenon. One light which is coming from the internal optical layer, another light which is coming from the ligand end. So when there is an interaction between the two binding partners, say suppose I am coating with a kind of a matrix, it is a protein A. When you dip into the sensor into the corresponding binding partner, example like monoclonal antibody, when human IgG starts binding to the protein A matrix, as you know that it is a concentration dependent, as the more molecule starts binding to the protein A matrix, then we can able to see there is a change in the spectrum, or I mean to say it is like a change in the wavelength. So the change in the wavelength is nothing but as the concentration increases, the change in the wavelength, it is corresponds to the optical thickness. The more the optical thickness, as we can see as the change in the wavelength, it indicates that the more the molecules are binding. So from this we can able to like determine the concentration. This instrument we can useful for the quantification purpose. Say suppose in case of the industry or any protein, if you want to do a quantification, directly we can take a known concentration of the standards, we can generate a standard curve, and from we can able to determine the unknown concentrations. Apart from that, the major useful of this instrument is to study the binding kinetics, where we will determine the rate constants like on rate, off rate, as well as the affinity constants. I'm going to demonstrate you kinetics interaction of the most monoclonal antibody to the protein A ligand. Here I am using biotinylated protein A as a ligand. At the concentration I am using a 10 microgram per ml. 10 microgram per ml I am putting into the 6 wells, and most monoclonal antibody I am using as an analyte. Here I am taking the starting concentration at a 25 microgram per ml. I am going to do a serial dilution with a twofold from 25 to 12.5, then serially I go for a twofold concentration around 6 data points. This is the plate map here. This is the buffer, the first column, and the second column I am going to put the 10 microgram per ml concentration of a biotinylated protein A, and the third column I am going to a buffer. It will be useful as a for a unbound biotinylated protein A get washed off here, and also this well I am going to useful for the dissociation purpose. And the last well I am going to put the analyte, that is a 25 microgram per ml serially diluted in all these 6 wells. This is the sample plate, 96 well plate. I am going to place in the sample compartment here like this. There is a click sound, it will come, firmly it fits into the sample compartment. Here this sample compartment, if you look at it, works on the dip and read, the BLI technology works on the dip and read method. Here we do not have any microfluidics devices. So to assist for a binding, we have a shaker. That shaker assists in the binding. The shakers works from 150 rpm to 1500 rpm, and also this sample plate have there is a temperature. We can work from the ambient room temperature like 23 plus 4 degree to the 40 degree centigrade. So here I am going to place the sensor compartment. So we pick up the sensors from the either column 1, column 2 or column 3, like anywhere. We can make it program according to the instrument. We are placing the sensor rack like this. If you look at this in the software, we have in a 40bio platform, the Biotechnology, we have a acquisition software as well as the data analysis software. Now I will show you the acquisition software, what are all the features it have. If I double click on this, instrument starts initializing. If you look at it moves in the XYZ direction, itself it initializes. Okay, now you can see that there is an initialization process is going on. It takes roughly around 30 seconds of time. So you can see in the software what the event is happening on the dialog box opened. Now it is showing that the instrument status, it is ready. So now we can start the experiments. When there is a acquisition software open, we can see there is an experimental wizard here. So here in the wizard we can see there is two major experiments, whether you want to do a kinetics experiments or the quantitation experiments. Now we are going to perform the kinetics experiments. I will go here clicking on this green arrow mark. So if you look at this, there is a page get opened here. If you see on your left hand side, there is a 96 well plate design. This is what we call it as a plate design. We have to mention in the plate design what things we had put it. So I had mentioned we had put a biotinylated ligand, buffers, as well as the twofold dilutions of the twofold dilution concentration of the most monoclonal antibody. Now we have to mention where all these things we had put into the 96 well plate. So I had put in the 96 well plate here, this column I put it as a buffer. I will right click it here, right click, I am showing this as a buffer. Here second column I had put a biotinylated ligand, that we call it as a load. Load is nothing but we are performing the biotinylated protein A immobilization step we are performing on the streptavidin sensor. Now the third column I had put here this one as a buffer. Once again unbound material get washed off here. Now we have to put analyte, we call it as a sample. So these are the sample IDs, we have to mention what we had put it here. We can put it this here just like a buffer. So what I will do is just I will copy it, then buffer, and once again here I had put a, once again there is a buffer, here I put a protein A as a ligand. Here as I mentioned the buffer, we can use any kind of buffer. Now in this experiment I am using a PBS buffer having a additive like a BSA, it is a 0.1% BSA, as well as it having a like a surfactant like a 0.02% of the twin 20. So this composition in a PBS at the pH 7.2. So I am here I am mentioning the mouse monoclonal antibody. I will use M small g capital G MIGG. Okay, I am using a 25 microgram per ml, and the molecular weight of the mouse monoclonal antibody is 150 kilodalton. It is automatically calculates it is a 166.7 nanomolar. Okay, just I will copy this one. What I will do, I will select this such well data, then it is a 25 divided by 2, then automatically last one I am put it as a 0 concentration. These are the concentrations 25, 12.5, 6.25, 3.125, 1.56 and 0.7 corresponding nanomolar concentration. Now we had in the, there is one more step, we call it as a assay definition. So these are the sum of the steps I am going to add here. There is a baseline by default, baseline just acquires the buffer. Now we are going next step we call it as a loading. Loading is nothing but we are immobilizing a 10 microgram per ml protein A onto the streptavidin sensor. So I am going to add here, then next step I am performing the baseline and I will say it as a OK. Then our instrument says that already there is a one more, first already we have a baseline, there is a exist, it is saying do you need a one more? Yes, I need one more. I will say OK. Then I will add one more step, we call it as association steps, we call it OK. Then second we call it as a dissociation step, OK. Now we have to tell instrument what is the first step. This is the first step, baseline. I am going here and double click on that. This is the first step, we call it as a baseline step. The second step we call it as immobilization, protein A immobilization, biotinylated protein A immobilization onto the streptavidin sensor. I will double click on this. Then the third step, baseline, once again unbound material get washed off here. Just I am clicking on this. Next step is the association step. This is the sample, this is the mouse monoclonal antibodies, two-fold diluted, 6 data points. Then the dissociation step I am performing a same buffer, that is the column 4. Okay, these are the what type of sensors we are using. I am using a streptavidin sensor. Now we have to say for an instrument how much time each step has to be acquired. The first step I require around 30 seconds, and the second step it's require around once again the 30 seconds. Third step it's good to have 60 seconds. Association I will perform for 150 seconds, and the dissociation I will perform for around 200 seconds. This is for this experiments, these are the conditions we already pre-optimize. So for this it is well established I am performing, and here I am performing the shaker speed at the 1000 RPM. If you look at the total time, it is roughly around 9 minutes. And now we have to say in the sensor compartment from where the sensor has to be pick up. Now I had put a sensor somewhere here at the third column in the sensor compartment. If you look at the next step, review experiment, in the review it is like that your sensor is picking from the third column, then the first step will be your baseline, second step your loading, third step your baseline, and the fourth step your association, then the fifth step your baseline. So run experiment, then where exactly you want to store the data. Here I am going to store the data in the desktop, have a IIT demo, I am creating this folder. Okay. Whenever you are performing the experiment, sensors has to be hydrated prior to the experiment. At least 10 minutes it is required for the hydrations. Say suppose your sensors are not hydrated, we can say in the software delay 600 seconds. 600 seconds is nothing but 10 minutes. After the hydration, the sensors, the instrument starts acquisition. In second option is like that during the the hydration, whether your sample has to be in the shaking conditions, because the instrument have a shaker, it will during the effectively mix. In some cases it is required, some cases not. You know that shaking sometimes enhances the aggregation. So we can uncheck this, and also I think prior to the experiment we already started a hydration of the sensors. Now sensors got hydrate, we can directly uncheck this, we can go for the experiment. And sample plate temperature, it is at the 30 degree, so already it is stabilized the plate. We can now go for the experiment. Here if you look at this, I opened the door for the demonstration purpose. In all the cases we have to close this door because stray lights affects the interactions. Now for the demonstration purpose, I'm keep opening the door. I will say go. Sensor is picking from here, from the sensor rack. It's now it is going to the sample plate. Sample plate, if you look at now, there is a shaker which is working at the 1000 RPM. So you could able to see there is a as when it dips into the sample compartment, you see there is a signal. So now this signal we are acquiring for the 30 seconds. The first step we call it as a baseline. Now it is going to a next column. It consists of a biotinylated protein at 10 microgram per ml. Now we can able to see there is a rise in the signal. This is what we call it as the immobilization step. The biotinylated protein A is binding to the streptavidin sensor. Now that is we are acquiring for around roughly a 30 seconds time. And it is around in less than 20 seconds we can able to see it is reaching to the 1 nanometer. The binding we are measuring in terms of the nanometer here. This is a good enough loading for to get the interaction studies. Now the moving to the next column where there is a buffer, PBS buffer, unbound material get washed off, and this steps is required for the to get a stabilized baseline. The baseline is now stabilized. We can go from here, we can say okay, it automatically goes to the next step, 30 seconds. Now the sensors are moving to the association step. Now here there is a biotinylated protein A interacting to your mouse monoclonal antibody. It is like a two-fold different concentrations. If you look at this, the graphs, this is how the curves are two-fold dilutions. This is the highest concentrations, this is the second fold, this is third fold, this is fourth fold. One at the bottom I had put the 0 concentration. So in a software we have a function like that. We can extend the current step, as well as if you want like it is already like a saturated, we can go to the next step. I will say go to next step. 100 seconds is good. The sensors went back to the buffer well, where exactly if there is a interactions happening, if it is a loosely bound kind of interactions, just come out from the sensor. If it is a very strong, then you can able to see there is a still kind of a straight line here. So if it is a straight line, then it indicates that it is a very strong binding. Once the dissociation step performed, the sensors once again re-rack back to the sensor tray, and the instrument, the robotic arm, moved to its original position. So now acquisition done. Now we have to go for the data analysis. There is a data analysis software. I am going to double click on the data analysis software. In the bottom of this, where exactly you have a data. So IIT B demo, I will go with this. Kinetics demo, I have a folder here. This I am double click on this. If you see that there is a page got opened, I will go to processing here. In the processing tab, we will see this the raw data. So what we are doing is we are subtracting. I had like one of the column I had put a 0 concentration. This column I will change to the reference well. This is what I am 0 concentration, because some of the artifacts from the buffer we can subtract from the data. So that is why I am going for the reference subtractions. On the left hand side, there is a here we have icon subtraction. We have different types of the subtraction systems. So I am using here one of the just only a 0 constant I am using for the subtraction. So reference well I am going for that. In the raw data I will go that I will select this association step. Then there is a align y axis. I am going to do a baseline. This is the baseline step for this particular baseline. I am going to align. Now I have to go for the process data. So if you look at now the data what we got, this is what the process data. Now we have to go for the analysis. There are the 6 data points we have here. In the analysis part we have association only, dissociation only, association and the dissociation. To calculate the rate constants we have to go always choose the association and the dissociation. We have different binding models like 1:1, 2:1 for the heterogeneous ligand. Mass transport effect, if there is any mass transport involving the interaction studies, we have a bivalent analyte 1:2 binding model. Depletion studies, yes. So local in the fitting model, apart from the what kind of model, we have another kind of a fitting. It is a local fitting as well as global fitting. Most of the times when you are performing for the interaction studies, we always go with a global fitting. So now I have to choose a global fitting. Global fitting with respect to color and the Rmax unlinked by a sensor. To perform a global fitting, we have to select all the samples, change to a one color. So I will change to a blue color. Now you can see this. Now is a 1:1 binding model. I have to try use entire step systems. I have to fit the curve. So now fitting is okay, and the Kd it is showing that around a 17 nanomole. Easily we can able to see the good fits through the with respect to the chi-square values as well as the R-square values. If you look at in this table, we have what is the Kd errors, K on with the K on errors, and if you look at the good fits, it is always when in case of the kinetics, the good fit comes it is a 0.95 above, and also chi-square should be less than 3. So from this indicating that it behaves a 1:1 binding model, it is supported by your R-square as well as the chi-square statistical parameters. Also we have graphs like XY graph, isoaffinity graphs as well as the steady state. Because in this experiments we not reach the steady states, we not able to calculate the steady states, but still software can pick up, and the Kd value it is determining it is around a 2.1 nanomolar, and also here also if you look at this is around roughly a 1.7 nanomolar. It is almost like a similar, very near to those values. Yes, it is a good fit, and we can export this into a printed versions like making a report. Here we have to say save report, then say export. We can take like a kind of a Excel sheet, what the parameters I had used for that, experimental summary, the sensor tray, where exactly we had put a sensors, the sensor data, what sensor we had used, sample data, how your sample plate design is, raw data, you can see the raw data, aligned data as well as the process data, how this we had subtracted using a reference well subtraction method, the graphs, the fitted graphs, the residual view versus the experimental view, the blue ones which are the experimental, red ones which are the theoretical. In the results tables, what is the where the sensor location, sample ID, the concentrations, response what we got, what is the Kd errors. So all entire details we can get it into the report. Now we successfully demonstrated the protein-protein interactions here with the protein A with the mouse monoclonal antibodies we used, and we got a very good data with the 1:1 binding model with the affinity is around 1.7 nanomolar range. So with this I'm concluding my demonstrations.
So, today we have witnessed the application of BLI to measure interaction between protein A and mouse monoclonal antibody. As demonstrated, the system monitored association of the analyte with the immobilized ligand and dissociation after moving the sensor to solution without the binding partner. The changes in interference patterns were quantified and used to determine the kinetic rate of binding and dissociation. Thank you.